Today we're going to talk about the structure of DNA molecules and the replication of DNA as well. First, DNA was isolated by Friedrich Mischer, and he named DNA nuclein, which gave rise to the name nucleic acids, because it was isolated from the nuclei of white blood cells. Later in 20th century, British scientist Friedrich Griffith discovered the process of bacterial transformation, in which external naked DNA is taken up by a bacterial cell, which in turn can change the cellular physiology. Griffith used two strains of bacteria Streptococcus pneumoniae. Rough strain, which was non-pathogenic, and the smooth strain, which was pathogenic because it had a capsule around bacterial cells. And this capsule protected bacteria from phagocytosis. When live S strain was injected into mice, mice died. When the live R strain was injected into mice, they survived, because R strain is non-pathogenic. Then Griffith generated heat-killed S strain. He inactivated bacteria with heat, and when this heat-inactivated S strain of bacteria was inoculated into mice, mice survived. So capsule alone, which was still there even after heat inactivation, was not the cause of death. Then Griffith mixed a live R strain and the heat-killed S strain. And when he did that, mice died. When Griffith recovered the bacterium from the dead mice here, he only recovered the S strain, which maintained its pathogenicity. Obviously, something from the heat-killed S strain changed live R strain into pathogenic S strain, and that something was DNA. Later, Avery, MacLeod, and McCarty isolated proteins, RNA, and DNA from the S strain. After that, they specifically degraded each component. They degraded protein, they degraded RNA, or they degraded DNA. And then they used each separate mixture to transform the R strain. Only when DNA was degraded, they did not observe any transformation. And all other combinations, whether RNA or protein, were degraded. That resulted in transformation. So they made a conclusion that the transforming principle was the DNA. To further prove the role of DNA as a storage of genetic information, Chase and Hershey performed a famous experiment of bacteriophage infection of bacteria. Bacteriophage is a virus that can infect the bacteria. In some cases, it is called a phage. It has a pretty simple structure, a protein capsid that surrounds a nucleic acid genome. During the infection, a phage injects its DNA into bacterial cells. Once the genome is inside the cell, phage will reproduce and eventually break out of the host cell, lysing it. One batch of phage that Chase and Hershey used was labeled with radioactive sulfur as 35, which essentially labeled protein capsids. Another batch was labeled with radioactive phosphorus, phosphorus 32, which labels DNA genome. They used each batch of phages to infect bacterial cells separately. You can see the labeled DNA and the labeled protein code. And then they separated phage capsids from the host cells. So it turns out if phage was labeled with sulfur 35, all radioactivity remained in the phage supernatant, but bacterial cells were not radioactive. However, cells that were infected with a phage that was labeled with phosphorus 32, the radioactivity was found in the pellet of bacterial cells. In other words, phage DNA that was injected into the cells carried information to generate more phage particles. This experiment is considered to be the final answer to the question which molecule stores the genetic information in the cells. Erwin Chargaff later started to investigate the composition of DNA by measuring the relative concentrations of 4 different bases found in the DNA molecule, A, T, G, and C. He discovered that the relative concentrations of these bases in the double-stranded DNA that comprises cellular genome varies from one species to another. However, it doesn't vary between individuals of the same species. He also discovered that the amount of adenine A equaled the amount of thymine T, and the amount of cytosine C equaled the amount of guanine G. In all species that he studied, the amount of purines A and G were equal to the amounts of pyrimidines T plus C. However, the ratios of AT pairs to GC pairs were different from species to species. This rules that the ratio of A and T are the same, and the ratios of G and C are the same, that the ratios of purines and pyrimidines are equals, were called Chargaff rules. Now let's change gears and talk about the building blocks of DNA, which are nucleotides. A nucleotide, shown on the bottom left of the page, consists of a nitrogenous base, a pentose sugar, and a phosphate. The name of the nucleotide is derived from the nitrogenous base that it contains. There are 5 nitrogenous bases found collectively in DNA and RNA. In DNA, you can find 2 pyrimidine bases, cytosine and thymine, and 2 purine bases, adenine and guanine. You can see the similarities between 2 purines and 2 pyrimidines. In RNA molecules, instead of thymine, you can find a pyrimidine base, uracil. The sugar that you can find in a nucleotide depends on the type of nucleic acid you are dealing with. If it's a DNA, it's going to be deoxyribose with the hydrogen at the 2' carbon. If it's RNA, it's going to be ribose with OH group at the 2' carbon. You can see that the base is connected to the first carbon, so-called 1' carbon of the sugar, while phosphates are attached to the 5' carbon of the sugar by the ester bond. Now, the structure that contains only base and the sugar is called a nucleoside. So the full name, the big name for a nucleotide will be nucleoside monophosphate, if only one phosphate group is attached to 5' carbon, nucleoside diphosphate, if 2 phosphate groups are attached to 5' carbon, and nucleoside triphosphate, if 3 phosphate groups are attached to 5' carbon. Between themselves, nucleotides are linked by so-called 5', 3' phosphodiester bonds. They formed between 5' carbon of the sugar in one nucleotide and 3' carbon of the sugar in another nucleotide. When you look at the DNA molecule, the DNA chain, on one end, there's going to be a free 5' phosphate because it's an end, so it doesn't form a bond with another nucleotide. The end with a free 5' phosphate is called 5' end. The end of the DNA chain with free 3' hydroxyl group, which you can see here, is called 3' end. The structure of the DNA was essentially discovered in the early 50s by a team of scientists, including James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Rosalind Franklin studied the X-ray diffraction to study the structure of the DNA, and you can see the famous image on the right. So Watson and Crick, which were working alongside with Franklin, used her data to suggest that the DNA is a double-stranded molecule that are twisted around each other, forming a right-hand helix. Purines and pyrimidines that are sitting in the opposite strands form complementary pairs. A, which is a purine, pairs with the pyrimidine T, adenine pairs with thymine, and G, which is purine, pairs with C, which is pyrimidine, guanine pairs with cytidine. That explains Chargaff rules, that the ratio of A and T's are the same and the ratios of G's and C's are the same as well. These pairs form hydrogen bonds between themselves, and you can see that AT pairs form 2 hydrogen bonds, while GC pairs form 3. So GC pair is essentially a stronger pair. The strands of the DNA in the double helix are anti-parallel, so 3' end of one strand faces 5' end of the opposite strand, and vice versa. The backbone, shown on the left image as the blue ribbon, is formed by sugars and phosphates, while the bases are stacked inside. If you pay close attention to the molecule of DNA, you can see that the twisting of the strands around each other makes this molecule slightly asymmetrical, resulting in the formation of the major and minor grooves. These uniformly spaced depressions on the DNA molecule serve as the sites of attachment for various proteins that bind to DNA. James Watson, Francis Crick, and Maurice Wilkins received Nobel Prize for the discovery of the DNA structure. However, Rosalind Franklin was not considered for that award because she died by that time due to the excessive X-ray exposure. Modern science relies a lot on so-called DNA sequencing, determination of the sequence of the bases in the DNA molecule. There are two sequencing methods, Maxim-Gilbert method and Sanger method. We're going to talk about Sanger sequencing method, which is widely used in a lot of sequencing applications today. His method, also known as D-deoxy chain termination method, uses special nucleotides, so-called D-deoxynucleotides, which don't have a free hydroxyl group at the 3' carbon at the end, the sugar. Since they don't have that free 3' hydroxide group, the DNA cannot be extended once a D-deoxynucleotide has been incorporated into DNA. So how is that sequencing method works? The sample of the DNA is denatured, strands are separated, and it is placed into 4 tubes. Each tube contains a primer, short DNA sequence that allows an enzyme called DNA polymerase to start synthesizing a complementary strand, a protein enzyme called DNA polymerase, and 4 nucleoside triphosphates that we've been talking about before. 4 of these guys. One with A, one with T, one with G, and one with C as a base. Each tube receives one of 4 D-deoxynucleotides: CTP, ATP, GTP, or TTP. And each of these 4 D-deoxynucleotide triphosphates carries a different label that fluoresces. When the reaction is over, each sample is subjected to electrophoresis, which separates DNA fragments, and then laser scanner reads the fluorescent labels within the electrophoresis and essentially can see the fragments of the different length based on the place of incorporation of D-deoxy chain terminator. So gel electrophoresis that we've mentioned is the process that separates DNA fragments based on their size. Samples of the DNA, you can see the image on the right, are loaded into the gel made of a polysaccharide called agarose. Due to the composition of the buffer, the DNA during gel electrophoresis has a net negative charge, and when electric current is applied, it moves towards the positive electrode. The smallest fragments move farther, so in this image, the smaller fragments will be at the bottom of the picture, while the heavier fragments will be closer to the well right here. The gel can be stained with a DNA-specific dye, ethidium bromide, which allows scientists to view separated fragments. Now, how is DNA organized in the cells? DNA exists in the form of chromosomes. In prokaryotes, there is a singular circular chromosome that is located within the nucleoid. The DNA in the prokaryotic chromosome is supercoiled, which means that in certain regions it's underwound and in some regions it is overwound from its normal state. That supercoiling that allows to package DNA tightly is regulated by a variety of prokaryotic proteins. In eukaryotes, there are several linear chromosomes, and by several, I mean from 4 to 72. Within eukaryotic DNA, negatively charged DNA is wrapped around positively charged histone proteins. That wrapping results in the formation of a nucleosome. The nucleosomes are then linked by so-called linker DNA, like beads on a string. That string of nucleosome is compacted into a 30 nanometer fiber, and these fibers further condensated into chromatin. There are two distinct regions within the chromatin of a eukaryote. The tightly packaged region of a chromosome is called heterochromatin, and the genes that are located within that region are not expressed. The less dense part of the chromatin is called euchromatin, and the genes found in that loosely organized euchromatin are indeed expressed. So now we're going to talk about the process of DNA replication. During replication, two strands of the DNA separate, and each strand acts as a template for the synthesis of the new complementary strand. So based on this explanation, you can see that the replication of DNA is conservative. However, when this was studied initially, nobody really knew whether the replication process was conservative, in which strands in the parental DNA remain together and the new molecule is completely new. It could be semi-conservative, where every double-stranded DNA after the synthesis contains one old and one new strand, or dispersive, where both DNA copies have interspersed segments of parental and newly synthesized DNA. To answer the question, to elucidate that the DNA replication actually follows semi-conservative model, Messelson and Stahl performed a famous experiment with DNA labeling by nitrogen. They started by growing bacteria E. coli in a medium that contained a heavy nitrogen isotope N15, which was incorporated into bacterial DNA. You can see the density of the DNA from the E. coli cultivated on that heavy nitrogen on the left panel. Then they cultivated E. coli on the medium that contained normal isotope of nitrogen, nitrogen 14, for several generations. They harvested cells after each generation, isolated DNA, and centrifuged that DNA at very high speed in so-called density gradient. Within the density gradient, the DNA will form a band according to its ability to float, according to its buoyancy. So, if DNA contained more nitrogen 15, it would be heavier, have higher density, and the band would be farther down the tube. So after one generation, what they discovered is that the single band, it was single band, but it was between the DNA of the cells that were grown exclusively on the nitrogen 15 and cells that were grown exclusively the nitrogen 14. So that immediately suggested that it's either semi-conservative or a dispersive replication. After 2 generations, they observed that 2 generations of growth with nitrogen 14 resulted in the formation of 2 bands. 50% of the DNA were located at the level of nitrogen 14 DNA, and 50% were located at the mix at the between 15 and 14. That unequivocally demonstrated that DNA replication was semi-conservative. So, as we mentioned before, each of the 2 strands serves as a template for the synthesis of a new strand, and the new strands are complementary to the parental ones. The process of replication begins at so-called origin of replication. Origin is AT rich. Presence of large numbers of A and T pairs, remember they are weaker, makes DNA strands easier to separate. The enzyme helicase binds to the origin of replication, that's helicase, and unwinds, or sometimes it's called unzips, the DNA strands, using energy to break down hydrogen bonds between the base pairs. Obviously, since this process requires energy, it also requires ATP. 2 Y-shaped replication forks are formed, and these forks are extended bidirectionally. You can see only the fork that is extended from right to left, but there is another fork, supposedly, that extends from left to right. So they extend bidirectionally as replication continues, and you can imagine that 2 strands, quote-unquote, want to go back together. To prevent strands from re-annealing, single strand binding proteins stabilize single-stranded DNAs and keep these strands open. Now, in what direction does DNA replication occur? Well, DNA polymerase, the enzyme responsible for the synthesis, can only add nucleotides in the direction from 5 to 3'. It also needs a free hydroxyl group at the 3' carbon of the terminal nucleotide, so it can add more nucleotide to the growing chain. To provide free 3' hydroxyl group for DNA polymerase 3, so it can start synthesis of DNA, cells use the enzyme called primase. Primase synthesizes a short RNA primer that provides this necessary 3' hydroxyl group to DNA polymerase. You may remember that bacterial DNA in the chromosome is supercoiled. Topoisomerase prevents supercoiling, sitting a little ahead of the replication fork to allow helicase to unzip the strands without any issues. So, DNA strands, as you remember, have opposite orientation. In this picture, the top strand, the blue strands, represent the maternal strands. It has 3' end on the right and 5' end on the left. The bottom strand has 3' left on the left and 5' end on the right. So, since DNA polymerase can only extend the new strand in the direction from 5' to 3', it will synthesize the strand that is complementary to top 3'5 parental strand in the continuous and uninterrupted fashion. The strand that is synthesized in that continuous and uninterrupted fashion is called the leading strand. The other strand, however, extends away from the replication fork. So DNA polymerase can only move away from replication fork. Therefore, the strand, the synthesis of the second strand is discontinuous. It is synthesized in a form of small Okazaki fragments, and it is called a lagging strand. If you look at the bottom illustration, you can see that there is a primer. DNA polymerase synthesized that Okazaki fragment, jumped back on another primer, and starts the synthesis of another Okazaki fragment. When it's going to be completed, DNA polymerase 3 will go back to the next primer and will synthesize it again. So if this is the continuous synthesis of the leading strand, the lagging strand is synthesized like this. These curved arrows represent the translocation of the DNA polymerase. In order for DNA polymerase 3 to not fall off the DNA molecule, special proteins called sliding clamps hold it in place. Now, we have RNA primers still attached, complementary to the DNA strand. So, how can you remove the primer? There is an enzyme called DNA polymerase 1, which has so-called exonuclease activity. It removes the RNA bases, RNA nucleotides, completely removes the primer, and the gap that is formed as the result is filled by the DNA nucleotides by the same enzyme, DNA polymerase 1. However, after that synthesis, there are nicks, gaps, between the primer replacement DNA and the previously synthesized DNA, for instance, in the Okazaki fragment. These nicks are sealed by the enzyme called DNA ligase. Ligase essentially facilitates the formation of the covalent ester bond between the 3' hydroxyl of one fragment and 5' phosphate of another. So this is the summary of the functions. The enzymes critical for DNA replication, you must know them. And this is the graphic summary, which shows you the synthesis of the leading and the lagging strand, and highlights the roles of individual enzymes such as topoisomerase, helicase, DNA polymerase 3 and 1, ligase, sliding clamps, single-stranded binding proteins. DNA replication in eukaryotes is not drastically different from prokaryotes. There are some differences, though. So in eukaryotes, unlike in prokaryotes, there are multiple origins of replication because the DNA is much larger, the size. The rate of replication is much slower, by about 10 times. There are 14 different DNA polymerases and like 5 in prokaryotes. It's a special enzyme called telomerase, which is critical, and we're going to talk about it. Primers are removed by different enzymes. In prokaryotes, it is DNA polymerase 1. In eukaryotes, it is RNase H. The elongation, the synthesis of the strand in prokaryotes is carried out by DNA polymerase 3, one enzyme, while in eukaryotes, it is carried out by polymerases alpha, delta, and epsilon, and the sliding clamp has a different name. It's called proliferating cell nuclear antigen. Another obstacle in DNA replication in eukaryotes is that the histones that the DNA is wrapped around, these proteins, have to be removed and then inserted back. Now, what is that telomerase and what are telomeres? So you may remember that the chromosomes in eukaryotes are linear. And you also remember that DNA polymerases can only add nucleotides in 5' to 3' direction. So in the leading strand, it's not a problem. Synthesis will continue until the end of the chromosome, and it will be completely copied. However, in the lagging strand, the DNA is synthesized in the short stretches, which we call Okazaki fragments. So when the replication fork reaches the end of the chromosome, primer doesn't have enough space to form on the 5' end, and the part of the DNA at the end of the chromosome remains unpaired. We call these fragments telomeres. So you can imagine that with each act of DNA replication, the telomeres will get progressively shorter. In other words, cellular division shortens the telomeres. Now, telomeres do not encode any particular gene. They're just repetitive sequences. For example, in humans, the sequence TTA-GGG is repeated up to 1,000 times in the telomere region. Essentially, telomeres protect the actual genes from getting deleted, because as cells continue to divide, telomeres shorten. The enzyme telomerase adds telomeres at the end of the chromosome. So telomerase, shown here in blue, contains two parts, a catalytic part and the RNA template. It attaches to the end of the chromosome, and DNA nucleotides that are complementary to the RNA template within the inside of the telomerase so this is the inserted DNA nucleotides, this is the RNA template. They are added at the 3' end of one of the DNA strands, which means when 3' end of that lagging strand is elongated sufficiently, DNA polymerase can now add nucleotides. Here's the DNA polymerase. It can add nucleotides complementary to the chromosomal ends, and the ends effectively are replicated. You can see that illustrated right here. So this DNA polymerase will be moving from adding nucleotides from 5' to 3', as we mentioned before. So you can imagine that telomerase is highly active in cells that divide often, such as germ and stem cells. Adult somatic cells which do not divide often have very low telomerase activity. Mice that are deficient for telomerase have tissue atrophy, depletion of stem cells, organ failure, which is not surprising because telomerase is essential for the cells that divide and essential for the maintenance and growth of organs. If telomerase is reactivated in such mice, telomeres extend, and it reduces the damage to the DNA and improves cellular function. Interestingly enough, cancerous cells have shortened telomeres, which isn't surprising, they divide fast. However, the telomerase is very active in these cells, and it is possible that the regulation of telomerase activity may lead to the discovery of the special treatments for the age-related diseases and cancer. Now, during DNA synthesis, which is highly accurate, mistakes can happen. For instance, a wrong base can be inserted. Most of these mistakes are corrected by so-called proofreading, or 3' exonuclease activity of DNA polymerase. For example, here, the G is inserted against A, which is incorrect. DNA polymerase itself will read and find that mismatch. And part of the DNA polymerase 3 that has that exonuclease activity will cut the phosphodiester bond, will release that nucleotide, the G, and will insert the proper one. So this is the proofreading. Mismatch repair is when wrong pair has already been kind of sealed within the freshly synthesized daughter strand. In case of mismatch repair, special enzymes, they remove the part of the strand with it, and the region that is excised is synthesized again. For example, in E. coli, adenine, one of the bases, is methylated after the replication. So, very much after the replication. So parental DNA strand will be methylated, but the newly synthesized strand does not have methyl groups, which means DNA polymerase can recognize the wrong bases only in the new strand that is non-methylated and successfully remove them. In eukaryotes, most likely, unsealed nicks are recognized in the improperly synthesized strand. For damaged bases, cells often use nucleotide excision repair. So during nucleotide excision repair, special enzymes cut damaged strand on both ends, and this is done by the enzyme called nuclease. The enzyme helicase, which is not shown here, removes the single strand of DNA, and then DNA polymerase replaces that gap with a correct DNA using another strand as a template. Finally, DNA ligase seals it, forming phosphodiester bonds between the new and the old DNA. This method is often used when exposure to UV light results in the formation of so-called pyrimidine or thymine dimers. An example in which this repair mechanism is defective is the condition known as xeroderma pigmentosa, sensitivity of the skin to the UV rays. Exposure to UV light results in the formation of pyrimidine or thymine dimers, and this damage is not repaired because nucleotide excision repair enzymes are defective. These thymine dimers distort the double helix, and that may lead to the problems during DNA replication, putting those people at the greater risk of skin cancer. Now, mistakes in the DNA are called mutations. So mutations are essentially the variations in the sequence of nucleotides in the genome. Mutations can be spontaneous, that are due to the unfixed replication mistakes, or they can be induced, which may result from the exposure to chemicals and radiation. There are two main types of mutations: point mutations and frameshift. Point mutations affect one base pair. Transition substitution is when a purine or pyrimidine is replaced by another purine or pyrimidine. For instance, A is replaced by G, or T is replaced by C. Transversion substitution, though, is when a purine, for example A, being replaced by a pyrimidine, for example C, or vice versa. Based on the effect on the protein sequence, mutations can be silent. They do not alter the protein amino acid sequence. You can see here, every amino acid is encoded by 3 letters called codons. We're going to talk about them soon. So the codon for GTA is, for valine, for amino acid valine, is GTA. The codon for valine is GTT also. So GTA or GTT, at the end of the day, it's still valine. Missense mutations result in the replacement of one amino acid by another. For instance, proline, encoded by CCC, is changed to threonine, encoded by ACC. Nonsense mutations result in the formation of so-called stop codon that terminate protein synthesis instead of an amino acid. Frameshift mutations are formed when a base is either added, insertion, or removed, deletion. So in this example, you can see that T and A are removed, and that leads to a complete change in the amino acid sequence of the protein that is now encoded by this new sequence. Another interesting type of mutation is called translocation, when the entire chunk of DNA is moved from one chromosome to another. It's important to understand that while mutations can change the structure and the function of the protein, they don't necessarily have to. Silent mutations won't, and even missense mutations, if one hydrophobic amino acid is replaced by another hydrophobic amino acid, may have no effect on the structure and the function of the protein.